today. Because they all got killed at where? In the vagina. Who created that craziness in the vagina? And is that? No, the bacteria. Remember we talked about this thing. Yeah, the acidophils. If you have too much acid in that vagina, the Y chromosomes die easily. They're just totally destroyed. Hence, new research are coming out that yes, it is the female's fault. I'm sure it started by the male side.
Of course it did.
But genetically, it's a 50-50 chance by the male. Right? Just looking at genetic profile. So, the dominant gene is what? Is the one that's always shown to you. Then the one that sometimes shows up, he said is called the recessive gene. And he made a very ambiguous statement. He said that it's a gene that not always is expressed. Because he's a scientist. Scientist never likes to make a conclusive statement. Because you never know. You don't want to be embarrassed 200 years from now. Just making claims like what? An egg and a sperm cell. One is the food and the other one is? It's a little guy. So, this is what he understood, and he wrote down all the observations, and he just did ratios, because he's a mathematician. Who came along and explained this, really? 60 years after he died, a guy came along and said, we can explain this a little bit better now. So 60 years, which means we're almost in the 1900s. What was that guy's name? His name is Punnett. But before we can even get to the Punnett kind of concept, what do we have to understand? If you are tall, as Mendel is stating, the understanding of spermatogenesis will tell you this, or oogenesis. Remember, if you are tall, your father gave you what? Putting this in color perspective again, and hence the reason why we always colored it last time. He said, your mother gave you a trait and your father gave you a trait. And Mendel is claiming, if mother gave you a tall and your father gave you a tall, what are you? You are a tall plant. If your mother gave you a short and your father gave you a short, you are short. So what does this represent for you? That's what you call the G1 cell. A cell that is in interphase. What happens? After the S phase, what are you? A G2 cell. What happened to your big T's?
Doubles.
It gets copied. What happened to your little t? It gets copied. And that little dashed line is joining them together, which is called the centromere. Remember that concept? Oh, wrong T's. So, what happens to this cell after the S phase? The little t's copy themselves, and the little t's copy themselves. And they undergo what? Meiosis 1. Because of meiosis, they line up in synapsis, which means that what? The big T's are not split. They're held together. The blues are held together and the reds are held together. Everybody understand that? And hence this is the reason why we call these replicate haploids. Did we not? Mm-hmm. Well, these were called replicate diploids. That's that, and that's that. So we should be really proficient at this now, hopefully. So what happens next? It's meiosis II. How many cells do you get at the end of
One.
that you call gametes? 4 for each of these guys, do you not? What's in this one? Little t, little t. Little t, little t. Which are your gametes? This is big T, big T. Big T, big T. And Punnett said I can show you this probability in a box form. Because there are 4 different types of gametes that are produced by each of the plants. And he labeled males on top and females on the bottom. So if one of these plants was male, let's say this one was male, and this one was female. What are the outcomes? They're all big T's. What are the females' outcomes? All little t's. And Punnett is just saying if I combine this, so in other words, if this was my sperm cell, it will eat up one of these eggs. And these are the only 4 possible eggs, are they not? One of them is an egg, the other 3 is a polar body. If this one happens to be the ovum, these 3 are polar bodies. If this one happens to be the ovum, these 3 are polar bodies. If this one happens to be the ovum, those 3 are the polar bodies. Everybody understand that? But regardless, who is the egg, I have to be able to show you all of them. All the possibilities. So, what does Punnett say? The first outcome is the sperm meeting up with this egg. That's my outcome. So you have 4 scenarios. Or possible ovum. Not all 4 of them are going to be an ovum, right? If one of them, if this one happens to be the ovum, then this is polar bodies. If this is ovum number 2, then the others were polar bodies, so you would have to only concentrate on this line. Everybody understand that? If this is ovum number 3, that is our possibility, and the others should be ignored. And if this is our 4th, and the other ones are all polar bodies, so what he's basically doing is giving you every possible outcome in a table. So, what is the only possible outcome that you see? They're all going to be big T, little t, 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 big T, little t. Everybody understand the outcomes? And he said what? The only type of genes that you can see here mixed, and we'll call it the genotype. What's a genotype? It's the genetic makeup of the organism. What are the genotypes that you see? Always expressed by 2 traits, two letters. One the dominant, and the other one being the other allele. Right? Then phenotypically means what? The physical appearance of these. So if you have a tall gene and a short gene, which is the dominant gene? The tall one was, so therefore the phenotype is tall. What was the probability of this outcome? It was 16 out of 16. So probability-wise, this is at 100%. Showing you that what? What Mendel's observation was true. Everybody understand that? Yes. Talk to me. Are you stretching? Think about it a little bit. Anyone else has a question?
So it's always most small.
Anytime, so one of them is a dominant gene, and Mendel stated that what? The tall is going to be the dominant gene. Why? It's always expressed, or mostly expressed. Okay? And he recognized the short to be the recessive gene. Okay? So this is a Punnett square. It's called a Punnett square because it's a square. Only problem with it is what? Your textbooks in the last 20 years. Stupid things are happening in biology. What is it? It's called political correctness, which is really dumb in science. What did they show you Punnett squares in your book? They don't show you a Punnett square, they show you a what? A Punnett diamond. Well, the Punnett diamond is not called a Punnett diamond, it's called a Punnett square, they write in the book. You get confused. Why? Because Punnett always put who on top?
Male.
Males on top, he said, and females on the bottom. And then, of course, some people said, oh, why is males always on top? Why don't we just put them on equal footing? Put the males on the right and the females on the left. So you would have to draw this table how? And then say what? The result is the same thing, but they said it's important that we show it this way. Not to be sexist. I have a problem with this, because your brain does not understand what? Diagonal lines. Hence the reason why when you draw diagonal lines, what does the brain automatically tell your body to do? Tilt your head. So it makes it first vertical and horizontal. My second problem with this is what? Why is the male on the right? Then, does that mean males are always right and females are always wrong?
Why the male genes is first as well. Big T, little t.
So, again, remember, left in Latin means what? Sinister. So therefore, females are evil.
Really?
So hence, if you're going to do this, just knock it off, please. Remember, biologists are not sexist. It's just the way that these guys originally wrote it. Okay? So, for those of you who feel that you need to do this, that's fine. But put it back into the square. How do you put it back into the square? We'll do the second example with that. So, this was the F1 generation. You had to know how the gametes looked. Did you not? So the F1 generation results. So this is the P1 and the F1 generation. I apologize. This is the P1 generation, is it not? Because these were the two parents.
Yes.
What happened with the child? He is purely what? Big T, little t. Which means this is in a G1 state of life. What happens in the S phase of life? He doubles the T's and the little t's. Means the G2 cell. And we will undergo meiosis 1 to become what? 2 cells. Again, it's a replicate haploid. Now you'll probably understand why we separate them as replicate haploids. Remember, what does it mean to be replicate haploids? All the big T's go on one side and all the little T's go on the other side. You don't have a big T, little t combination here, because the way they are split up protein-wise. So what are the 4 types of gametes? 2 big T's and 2 little t's. And this is the meiosis 2 results. Everybody understand this? Remember, on the next exam, you have to draw all of these flows.
Yeah.
Yes, you do. So speak now, forever hold your peace, because your textbook really doesn't talk about it. They assume that you understand this now.
Okay, so when he created the F phase,